Hello and welcome. Now in this lecture we are going to discuss NGS raw data acquisition and the hierarchical organization of sequencing data in the NCBI SRA database. There are many ways to obtain NGS data. However, the two most common approaches are obtaining raw NGS data from sequencing service providers or companies and downloading public raw NGS data from public databases. Now let's start with the first approach, obtaining raw NGS data from sequencing service providers or companies. Raw next generation sequencing or NGS data can be obtained directly from commercial sequencing service providers, genomic companies, or institutional sequencing facilities after completion of sequencing. In this approach, biological samples are submitted to a sequencing laboratory where library preparation, sequencing, and primary data processing are performed using sequencing platforms such as Illumina, Oxford Nanopore Technologies, or Pack Biosystems. In Bangladesh, several organizations and companies provide NGS and genomics related sequencing services, including Child Health Research Foundation or CSRF, ICDDRB Genome Center, and Invent Technologies Limited. These organizations provide services such as whole genome sequencing or WGS, 16S rRNA Amplicon sequencing, shotgun metagenomics, RNA sequencing, and bioinformatics analysis. After sequencing, the service provider generally supplies the raw sequencing output through secure cloud storage, FTP servers, external hard drives, or downloadable web portals. The raw data are commonly delivered in FASTQ format, which contains nucleotide sequences together with their corresponding quality scores. Depending on the sequencing platform and service package, additional files such as BAM files, BCL files, metadata sheets, and quality control reports may also be included. Researchers typically receive single-end or paired-end FASTQ files, sample metadata and run information, sequencing quality control reports, and adapter trimming or demultiplexing results if included. This approach is widely used because it enables researchers to generate high-throughput sequencing data without requiring in-house sequencing instruments, computational infrastructure, or advanced technical expertise. Now let's move to the second approach, downloading public raw NGS data from public databases. There are several public databases and repositories that provide access to raw NGS sequencing data. However, one of the most widely used and important databases is the NCBI Sequence Read Archive, also known as SRA. So we are going to discuss the NCBI Sequence Read Archive and understand the hierarchical organization of sequencing data within the SRA database. So what is the SRA database? The NCBI Sequence Read Archive or SRA is the largest publicly available repository of high-throughput sequencing data. It stores raw sequencing data and alignment information from all branches of life, including genomic studies, transcriptomic studies, metagenomic studies, and environmental sequencing studies. The SRA database is a part of the International Nucleotide Sequence Database Collaboration or INSDC. This collaboration includes the NCBI Sequence Read Archive or SRA, the European Bioinformatics Institute or EBI, and the DNA Data Bank of Japan or DDBJ. A very important feature of this collaboration is that data submitted to any of these organizations are shared among all three databases. Let's see methods to download SRA data. We can download raw sequencing data from the NCBI SRA database in multiple ways. Among these, three commonly used methods for downloading raw sequencing data are direct manual download from the run browser of the SRA database. Second is using preface command followed by the FASTRQ dump command from the SRA toolkit. This method is generally faster and recommended for large datasets. And the third is using only the FASTRQ dump command directly without using preface fast. This method is generally slower for large datasets. Now, before starting the methods to download raw sequencing data from the NCBI SRA database, let's first understand how sequencing data are organized in the NCBI SRA database. When researchers submit sequencing data to the NCBI SRA, the data are arranged into four important levels. These are Bioproject, Biosample, SRA experiment and SRA run. Understanding these terms is extremely important for downloading and analyzing public sequencing datasets. Let's first look at the overall hierarchy. Bioproject contains biosamples, biosamples contain experiments, and experiments contain runs. And these four database levels form a hierarchy. Bioproject is the overall study. It represents the complete research project. Biosample is the biological specimen. It describes the source of the sample used in the study. SRA experiment represents the sequencing library. It defines how the sequencing was performed on a sample. And the SRA run contains the actual sequencing reads. This is the raw data generated from the sequencing machine. So the study is organized from the highest level, Bioproject, down to the lowest level, which is the run, containing the raw sequencing files. Now let's understand each level one by one. First is NCBI Bioproject. A bioproject is a collection of biological data related to a single research initiative originating from a single organization or a consortium. It provides user a single place where all data generated from a particular study are organized and linked together. In simple terms, a bioproject represents the entire research project or study. It contains the overall description of a scientific initiative and is usually associated with multiple samples and multiple datasets. It describes the scientific objective, the organism being studied, and the overall scope of the project. Think of a bioproject as the umbrella folder that contains everything from one research study. Here is an example. In this example, researchers are studying the gut microbiome of diabetic patients. The entire study receives a bioproject accession number such as PRJNA 900001. Now let's move to biosample. A biosample describes the biological source material. Each physically unique specimen is registered as a single biosample with a unique set of attributes. In simple terms, a biosample represents the actual biological material that was collected. It answers questions such as what sample was used, from which patient it was collected, from which tissue it came, or from which environment it was obtained. Each collected specimen receives a unique biosample accession number. As we can see here, it is the biosample accession number for patient 1 stool sample. This is the accession number for patient 2 stool sample. So, if we collect stool samples from two different patients, each sample is treated as a separate biosample. Now let's move to SRA experiment. An SRA experiment represents a unique sequencing library and its sequencing setup derived from a biosample. This is one of the most important components in the SRA hierarchy because it defines how the sequencing was actually performed. Simply, the SRA experiment describes the experimental design used to generate sequencing data. It includes library preparation name, sequencing platform or instrument, sequencing strategy, source material, selection method, and read layout, that is single end or paired end. For example, this SRA experiment represents an Illumina paired end 16S rRNA sequencing of a V3-V4 Amplicon library. Now let's understand it with a simple idea. From a single biosample such as stool sample, researchers can prepare different types of sequencing libraries. For example, RNA-seq library, whole genome sequencing library, or 16S rRNA Amplicon library. Each of these library preparations is treated as a separate SRA experiment. Now let's understand an important concept. One biosample can have multiple SRA experiments. Why is that possible? Because the same biological sample can be sequenced using different sequencing strategies. For example, from a single patient 1 stool sample, researchers may perform RNA-seq, metagenome sequencing, and 16S rRNA Amplicon sequencing. In this example, we can see that the same biological sample is used to create 3 different sequencing libraries. Therefore, 3 different SRA experiments are generated, as we can see here. Now let's look at an example of SRA experiment metadata. It may include the sequencing instrument such as Illumina NovaSeq 6000, the sequencing strategy, that is Amplicon, the source, that is Metagenomics, the selection method like PCR, and the read layout, which is paired end. Now let's move to SRA run. An SRA run contains the actual sequencing output files generated by the sequencing instrument, and it is linked to a specific sequencing library, which is the SRA experiment. For example, these SRA run accessions correspond to the actual sequencing read files, usually in FASTQ format. Now let's understand an important point. One SRA experiment can contain multiple SRA runs, but this does not always mean biological replicates. Here is an example of SRA experiment with multiple runs. Multiple runs can exist for several technical reasons. For example, sequencing was performed across multiple lengths, sequencing was repeated to increase coverage, additional sequencing depth was required, or technical replicates were generated. Here we can see an example. Now let's look at a very common situation found in modern sequencing datasets. In many modern datasets, especially with high throughput Illumina sequencing, we commonly see one biosample, one experiment, one run. Especially when sequencing depth is sufficient in a single run, only one flow cell or sequencing lane is used. Sequencing is performed only once, and newer high throughput sequencing instruments are used. Here we can see an example of this. This type of organization is extremely common in RNA-seq datasets, Amplicon sequencing datasets, small bacterial genome datasets, and many Illumina sequencing datasets. Modern high-throughput sequencing often produce enough data from a single sequencing run. Therefore, one biosample commonly corresponds to one experiment and one run. Now let's understand this hierarchy diagram carefully. At the very top, we have bioproject, and this is the bioproject ID, which represents the entire human gut microbiome 16S rRNA sequencing study. Think of the bioproject as the umbrella research project that contains all samples and sequencing data from the study. Now from this single bioproject, we can have multiple biosamples. In this diagram, we see 3 biosamples. Each biosample represents an individual biological specimen. In this case, human fecal samples collected from different individuals. Now look below each biosample. Each biosample can generate multiple SRA experiments. For example, under this biosample, there is 2 SRA experiments, as we can see here. Why multiple experiments? Because the same biological sample can be used to prepare different sequencing libraries. For instance, one library may be a 16S rRNA V4 Amplicon library, as we can see here, while another may be a shotgun metagenomics library. Each unique sequencing library and sequencing setup receives a separate experiment accession. Now look at the bottom level. Each experiment can contain multiple SRA runs. For example, this SRA experiment contains these 3 SRA runs. These runs represent the actual raw sequencing data files generated by the sequencing machine. Multiple runs actually occur because sequencing was split across multiple lengths, sequencing depth was increased, or technical sequencing replicates were generated. Importantly, multiple runs don't usually mean different biological samples. They usually belong to the same sequencing library. So the hierarchy flows like this. Bioproject contains multiple biosamples. Each biosample can have multiple experiments, and each experiment can contain multiple runs. Although this diagram shows multiple runs under some experiments for illustration, in real-world datasets, many experiments actually contain only a single run. The important point is that an experiment represents the sequencing library, while a run represents the actual sequencing output files. Now let's look at the 3 major international sequencing databases. Those are the National Center for Biotechnology Information in the USA, the European Bioinformatics Institute in Europe, and the DNA Data Bank of Japan in Japan. These databases exchange sequencing data with each other as part of the International Nucleotide Sequence Database Collaboration, also known as INSDC. Although the sequencing data are shared and synchronized internationally across these databases, the accession prefixes used by each database are slightly different. As a result, the same sequencing dataset may appear in all three databases, but with different accession prefixes depending on the database system. Common prefixes are shown here. Bioproject accessions identify the overall study. The accession prefixes are slightly different across these three international databases. For example, PRJNA is used by NCBI, PRJEB is used by EBI, and PRJDB is used by DDBJ. For instance, NCBI, EBI, and DDBJ bioproject may look like this as shown here. Biosample accessions identify the biological specimen. The accession prefixes differ slightly among these three international databases. For example, SAMN is used in NCBI, SAMEA is used by EBI, and SAMD is used by DDBJ. Here we can see the example of different biosample accessions. Experiment accessions identify the sequencing library and sequencing setup. Again, the accession prefixes vary among these three databases. SRX is used in NCBI, ERX is used in EBI, and DRX is used by DDBJ. For example, here we can see the different SRA experiment accessions for different databases. The RAN accessions identify the actual raw sequencing data files. The prefixes are also different for RAN accessions. SRR is used in NCBI, ERR is used in EBI, and DRR is used in DDBJ. Here we can see the example of different RAN accessions. You may also encounter study accessions such as SRP, ERP, and DRP. These are SRA study identifiers linked to sequencing projects. The prefixes also differ among the three international databases. SRP is used in NCBI, ERP is used in EBI, and DRP is used in DDBJ. Here we can see the example of different study accessions. Now an important question arises. What is an SRA study? And SRA study is a high-level sequencing project record within the sequencing read archive system. It groups together samples, experiments, and runs that belong to the same sequencing study. In practice, bioproject and SRA study are often very closely related, and for many users they may appear almost equivalent. However, they are technically different database objects. Historically, the SRA database introduced SRA accessions first using SRP, ERP, and DRP as mentioned earlier for different databases. Later, Bioproject became the broader umbrella database used for organizing biological projects across NCBI resources. Today, most SRA studies are linked to a bioproject accession. So to summarize, understanding the SRA hierarchy is essential for navigating public sequencing databases and for downloading the correct datasets for bioinformatics analysis. In this lecture, we learned about the major approaches for obtaining raw NGS sequencing data. We discussed how sequencing data can be obtained from sequencing facilities and service providers and from public sequencing databases. We also learned about the NCBI Sequence Read Archive or SRA, and we understand how sequencing data are organized into bioproject, biosample, SRA experiment, and SRA run levels. Finally, we explored the accession systems of major international sequencing databases including NCBI, EBI, and DDBJ. In the next lecture, we will explore the methods for downloading sequencing data in detail. We will also demonstrate the download procedure of raw NGS sequencing data step by step and learn how to retrieve real sequencing dataset efficiently for bioinformatics analysis. That's all for now. Thank you everyone.